Okay, so we were pretty much done with most of the notes. We're just going to go ahead and finish out with cloning organisms. So you've probably heard something about this already, but know that clones are groups of organisms that are genetically identical to one another. So they are derived from a single parent cell or some single organism. This does happen naturally in humans. If you've ever met an identical twin, those are technically clones. But you might have heard about some of these in the news. So, like, first one that's probably the most famous was the first mammal cloned, and that was Dolly the sheep. She was cloned in 1997. But there's also been other instances. So here we have a carbon copy. So this was the first cloned pet, the first cloned cat. And ever since this technology has been established, it's kind of become a little bit more commonplace. So there are places that you can go, and if you have an extra $50,000 laying around, you can pay them $50,000 to clone your pet. So these two dogs are clones of one such buyer. So yeah, save your pet's fur in case you want to clone your animal. All right, so there are instances of natural cloning that occurs in animals. So here, one of them is the hydra. So the hydra will sometimes form these buds that you can see on either side. And as the buds grow, they're going to turn into a genetically identical new hydra. So hydras is an example of natural cloning through budding. But you also have things like planaria. So planaria is just a kind of worm, and you can clone these animals just by cutting. So here's a planaria that's about to have a really terrible day. So, okay, this planaria's worm is getting cut into 4 pieces, but don't worry about it because he's going to be just fine in a minute. In fact, I don't think this guy even really takes a second to contemplate what's happened to him. He just sort of keeps swimming along. And so each of these individual pieces is going to become a genetically identical clone. So if we wait about 2 weeks, you can see that after a while we start to form eyes on each of the 4 separate pieces, and then it'll just sort of grow separately. And then within 2 weeks, you have 4 genetically identical individuals. The other thing that in animals that can give you natural clones are like aphids. So I don't know if you've ever been out to a dandelion, you see these tiny green insects all over the dandelion. So those are aphids, and they can actually produce offspring via eggs that were generated from mitosis instead of meiosis. So for the aphids, they will just use an egg that is still diploid and make a clone of itself. So that aphid, she don't need no man. She can make babies on her own, clones of herself. So these are some examples of natural cloning in animals. So if I were to ask you for these, be able to rattle these off, we got hydra through budding, planaria that can be cloned by cutting, and aphids that can reproduce clones via these oocytes that are generated by mitosis, not meiosis. Plants can also be cloned naturally, and you've probably done this yourself. So probably the most, common example is stem cutting. So you can go into someone's yard, you see a plant, you can like it, you can cut it off at the stem, and then just replant it in some water, and it will regrow roots. And that plant will be a clone of the original one that you took the stem cutting from. The other one that you've probably seen a lot is like garlic bulbs. So if you take one of these individual clones, these are all identical. These are basically like, octuplets that are genetically identical to one another. So I can take one clone, plant it, and it's going to sprout leaves just like it has here, and regenerate a whole new bulb. Again, those are all genetically identical. So that's a way that you can clone garlic. Another thing that you might see are like strawberry plants. So if you ever look at a strawberry plant, they're going to get these long stems that grow out at the top, and these are called runners. And so the runners will have these little plants called plantlets that grow off on the side of them and develop their own roots. So these can be cut off, and those are going to be genetically identical but completely independent plants. So this is another way that we have natural cloning in plants. The strawberries with their runners, which generate plantlets, and each of these plantlets here and here is genetically identical to the original plant. Okay, so there are some artificial methods that we can use to do cloning. So remember, probably from your reading, that cells and early embryos are this word called pluripotent. And what that means is that they have the potential to become plural types of cells. So they have multiple options of each of the cells that you see in here. It can develop into many different cell types. That's where that terminology comes from. Pluripotent has potential to become many different cell types. So if you split the embryos, physically they can be split, you're going to make genetically identical individuals. So they use this to make clones in cows. But this is also what happens when you have twins. So except that embryo splitting happens naturally, and you have those genetically identical individuals. So this is what it actually looks like. These are xenopus eggs, so they're being split in half. And so this one's going to be split in half, and this one's going to be left alone, and you're going to see that they're going to develop just like normal little tadpoles, same as this guy over here, they're just smaller. So these two are identical to each other because they've basically been made twins. Okay, so that's embryo splitting. That's an artificial method of making clones in animals. So the one that you're probably more familiar with, that has more of the hoopla surrounding it, is the cloning via something called somatic cell nuclear transfer. So what's different about this is that you're using donor nuclei from an adult. So that's the donor. You're not starting with an embryo. You're starting with cells that are coming from an adult. And this was first pioneered by a man named John Gurdon in frogs. So when he was originally doing this, he was doing this in frogs. So the first thing for somatic cell nuclear transfer is that you're going to want to get your donor nuclei from some sort of adult differentiated tissue. So what differentiated means is that it's in its final form. So like it's probably a skin cell from the cat, or maybe a cheek cell from the sheep. Maybe it was a blood cell isolated from the blood. Point is, it's in its final form. This is different from an embryonic source. The original nuclei that you're going to be cloning from is from an adult and from final tissue, not anything embryonic. So that's step one. You get your donor nuclei from your differentiated adult tissue. After that, you need to start prepping some embryos for this. So the first thing you do is you get embryos, and you're going to UV irradiate them. And then what that's going to do is destroy the embryo DNA, because remember, you want to make a clone of the animal over here. So when you're getting your embryos ready, you don't want any of the embryonic DNA that was originally in this egg in there. So you're going to see them flashing a blue light, and that's the UV light that's destroying the DNA that's in these embryos. And then you're going to see them use this needle to take out the embryo. So let's go ahead and look at that. Okay, so you're going to see the blue flash of light, and you're going to see a bright blue dot. That there is the nuclei in the egg, and so you're going to see them reposition it, get the needle, and they're going to take out that embryonic nuclei in DNA, which they've already destroyed, and they're probably going to check it again just to make sure it's really out and it's really gone. Sure enough, it's done, so then it's done on to the next one. So again, they're going to flip it around, they're going to UV irradiate it, that's what that blue light is. She's looking for the nuclei, and you can kind of see it here. And then they're going to, again, use the needle to get it out of the egg cell. So they'll just prep their embryos like that. So once they have their embryos are ready, they're going to take the donor nuclei that they got from over here, from either the cat or the sheep or whatever organism they originally isolated from, and now they're going to put them back into the embryos that they cleared of the embryonic or of the egg DNA over here. So they get rid of the original egg nuclei, and they're going to put in the donor nuclei that they got from the adult sheep or the adult cat or whatever it is that you're cloning. This is the nuclei that's going in at this point. So that's the third step. So nuclei from the donor gets injected. And this is a much easier process, as you can see. So once you have your egg and your donor nuclei together, what they do is they actually shock it. They apply an electric shock, which fuses these two cells together, and then you can transfer those embryos into some sort of surrogate mother. And when they have the babies, they're going to be clones of whatever you started with here. So if I wanted to clone my cat, I would have taken the nuclei from the donor cat, and I would have clones of whatever I started with. Same with the sheep, right? So these babies have no genetic relationship to these surrogate mothers because all of their DNA was taken from the donors. These are genetically identical, so 100% match. So that is somatic cell nuclear transfer. So these are going to make great test questions. I'm going to just ask you to outline somatic cell nuclear transfer. You'll have to be able to tell me all of these steps. Okay. So the most famous example of clones, again, is probably Dolly the sheep. So she was the first mammal that was cloned, and her big claim to fame was that she lived about 6.5 years before succumbing to a lung disease typical of older sheep. So the other thing that was a big deal is that she was able to have a baby, and so people were pretty excited about that. Okay, so that is it for our cloning video. So go ahead and get your booklets all the way filled out and get started on those practice problems.